Welcome to the Biologicals, Are They a Barrier or a Game Changer webinar. Today, we're very excited to have both speakers with us today. We'll introduce them in a minute. But before we begin, some housekeeping items. This webinar will be recorded and be available after the live stream. It will be emailed out along with the survey and on Field Advisors YouTube channel. CCA credits are available. If you are watching this webinar live, you will get a QR code will be available at the end of the presentation. If you need CCA credits after watching the recording of this webinar, we recommend self-reporting. If you have any issues with self-reporting, you can please email myself or contact Field Advisor for assistance. If you have any questions throughout the webinar, please put them in the chat, and we will answer all the questions and have answers for those questions at the end of the webinar. So now I'm going to introduce the presenters. I want Mallory, will you please tell us your name? Because I do not want to butcher it, sorry.
Absolutely. Dr. Mallory Choudoir.
Thank you so much. She is an assistant professor and soil microbiome extension specialist at North Carolina State University. Her research focuses on the interactions between soil microbes and plants, with an emphasis on leveraging microbial communities to enhance crop productivity and sustainability. She is dedicated to bridging the gap between cutting-edge research and practical applications in agriculture. And then our second speaker is Dr. Laura Lindsey, and she is a professor of soybean and small grains at The Ohio State University. Her research and extension programs focus on improving soybean and wheat production through agronomic practices and seed treatment technologies. Dr. Lindsey is known for her work on field trials and data-driven recommendations to enhance crop yields and support farmer success. She is involved in Science for Success, a collaborative multi-state initiative led by land grant university researchers and extension specialists across the United States. So first off, we'll hear from Mallory, and we'll let you start off. Thank you.
Fantastic. Thank you. Okay, this morning I am going to discuss some essential best practice tips and considerations for integrating microbial biostimulants into soybean production or other sort of crop production systems. So my research program and also the use of biologicals in agriculture is founded on the critical relationships between diverse microbial communities living in the soil, within plants, outside of plants, and the beneficial agricultural activities that these communities carry out related to soil nutrient cycling, plant growth promotion, and also pest and disease control. If we want to think about some specific activities that beneficial microbes provide to plants, and these microbes, again, are diverse communities living in the soil, within the roots, on top of leaves, and also in the air and within water. And these microbes provide functions such as enhanced nutrient and water access, increasing nutrient use efficiency for a plant, improving or promoting plant development through plant hormone production, increasing abiotic stress tolerance of a plant, and also disease suppression. So given all of these activities, we are in a current landscape of immense choice and possibility and market value. This is an image of a subset of the companies and industries involved in the biological landscape from 2023, right? So these product offerings are growing, and it is a really challenging market to navigate for a number of reasons. So in the next few minutes, I want to break a few things down. So we can classify biologicals in a few broad categories. Biostimulants, this is a very broad term. We do have a definition from the Plant Biostimulant Act of 2023. So this is a substance, a microorganism that when applied to seeds, plants, the rhizosphere, stimulates natural processes to enhance or benefit nutrient uptake, nutrient efficiency, tolerance to stress, or crop quality and yield. So for the most part, these products, biostimulants, are not regulated. They are regulated at the state level, similar to fertilizers. There is a new biostimulant program launched by the Fertilizer Institute, which is an effort to have some evidence-based data for efficacy and safety. However, there are just a handful of these biostimulants certified, and it's a voluntary option at the time. On the other side, biopesticides are highly regulated. So these are naturally occurring substances that control pests or microbes that also have biocidal activities or other plant incorporated protectants. And this is, again, defined by the EPA and highly regulated. So for the most part, for the rest of this talk, I'm going to focus on biostimulants. So what are these biostimulants? Again, this is a broad range of products that can range from microbials, so living organisms, extracts of other living organisms, including plants, like we see a lot of seaweed extracts as biostimulants, and then other biologically derived products such as organic acids and fulvic acids. Just to note, there are certain biostimulants that are not themselves biological, so some of these trace nutrients, but they could potentially be derived from biological organisms. So again, just to distinguish between a living organism, a microbe, and a product derived from a living organism, such as an extract or a compound produced by a living microbe. Many of these are interchangeable technologies contributing to the confusion. So biofertilizers are sometimes called bacterial inoculants or cultures or bioformulations, inoculants. So it's important to understand all of these words that we're using to describe these products. Important distinction is natural versus synthetic. So these natural products, again, are from living organisms as opposed to created in a laboratory. And then one final distinction that is getting a lot of traction with the innovational strategies, including CRISPR gene editing. We hear a lot more about products that are gene edited. This includes both plants, but also microbes. And this is distinct from genetic modification, which includes inserting a foreign piece of DNA into an organism, whereas gene editing is related to modifying the existing genes that are in that organism. And then again, these regulatory conversations are underway. This is an image looking at the regulatory global landscape, targeting plants that are GM versus genetically edited, but similar conversations are happening related to microbes. So to put all of this together in a brief illustration, I like to think of these products as biostimulants, which can be microbial, so a living microbe, or something derived from something that was once living that stimulates natural and beneficial processes, wide range of activity, and mostly non-regulated. Biopesticides, on the other hand, have a very specific activity of controlling pests, and these are highly regulated by the EPA. Okay, so given that background, I want to move into outlining 10 essential considerations and best practice tips for using microbial biostimulants. Again, focusing on microbial biostimulants, however, these tips would apply to biopesticides or other non-microbial biostimulants. And this perspective comes from my years of research as a soil microbial ecologist. And also, I spent a few years as an industry scientist in a biotech company developing these microbial products. So that's sort of the background I'm coming from. Okay, tip one, reread the label and then reread the label again. At the end of the 10 tips, we'll go through some label examples. The things you're looking for are the guaranteed analysis. This will say the percentage of nutrients or other products, including microbes. Is this product a single microbial species or a strain, or is it a group of microbes, a mixed community? And then also the cell or dosage number. There will be storage and handling instructions, safety and compatibility, and importantly, application instructions. 2, select your products carefully. Be cautious of silver bullet solutions and big claims, right? There's a rapidly expanding market. So being cautious of claims that are wild and silver bullets, I think can really help hone in on products that are worth testing. Read the label. Certainly, there are a number of economic costs and return on investment considerations. We'll talk a bit more about this later. These products are typically crop or stress specific, and it's also important to identify your goal, if that is yield increase or potential replacement of other inputs. And we really want to utilize these products under optimal conditions to maximize the potential benefits. Determine or infer the mode of actions, right? So microbes provide a number of beneficial activities. So this mode of action can be general or it can be very specific. So multiple ecological functions are also possible. An example of a specific mode of action would be a microbe that is a biological nitrogen fixer, so providing nitrogen to the plant. Phosphorus solubilizing activity is also quite common. That would be another specific mode of action. And there are other plant beneficial microbes that have more general beneficial relationships that involve a number of potential specific mode of actions, including mode of actions can also be inferred if the microbe is a plant endophyte, so living inside plant tissues, or a rhizosphere microbe living within the root system. Number 4, follow storage and handling recommendations. This is pretty straightforward, but really important. We typically want to store these products in cool, dry, well-ventilated locations. Remember, these are living microbes. No extreme temperature, less than 80 degrees Fahrenheit about, usually above freezing. We want to keep away from direct sunlight, and for the most part, these are single use and keep sealed to prevent contamination. There's microbes everywhere, so when a bag or a jug is opened, unless you're in a sterile environment, it is going to be really challenging to prevent other microbes from floating into that product bag. And then be mindful of the expiration date. I certainly eat things from my refrigerator that are past the expiration date. However, there will be loss of viability and increased risk of contamination well past expiration dates. Identify potential incompatibilities. For the most part, microbes that are seed treatments are compatible with typical fungicides and insecticides. However, it's always good to test this. Sorry, not test it. Confirm this if there are specific incompatibilities. Sometimes mixing with fertilizers or other nutrients may alter microbial activity. And then depending on the formulation and the microbe, changes of moisture, pH may also influence viability and activity. One standard recommendation is to not use chlorinated water. So use non-chlorinated water, minimize chlorine. That is a strong microbial biocide, and most microbes are not compatible with chlorine. Ensure correct application mode, rate, and timing. These formulations are typically a liquid or a powder. Some of the powders need to be reconstituted in non-chlorinated water. Application will also determine your mode. So if this is a seed treatment or in furrow or a foliar application, again, we want to apply these in conditions that aren't too dry or too wet, or we risk losing viability of the microbe or literally washing the microbe away. And a good standard recommendation for liquid applications is at least 5 fluid ounces per 100-pound seeds. There's a lot of conversation about the utility and the efficacy of these products, and I encourage you to calculate and consider ROI possibilities broadly, right? So we certainly need to prioritize our cost of the product, the cost of special equipment, and time. But in this landscape, there's a possibility to consider things beyond crop yield and quality, such as reduced fertilizer inputs. So replacing nitrogen, I think, is a really exciting opportunity for these products. There's also crop protection. So utilizing these products in production environments that are prone to extreme weather events or increased environmental stressors such as soil salinization. And there's also benefits beyond our fields that are challenging to quantify related to environmental quality and water quality. So something to consider. Prioritize local performance data, right? So we really need unbiased third-party results. Be cautious of the side-by-side treated, untreated plots. We have all seen those before. They're super impressive. However, the rigor often of the data collected from that is not super strong. Again, small plot trials, and I do a lot of small plot trials. They're important for certain things, but they really have limited power for observing small effect sizes. Highlighting the importance of on-farm trials. In North Carolina, we have the On-Farm Trial Network, which was inspired by the Illinois On-Farm Trial Network. And then again, multiple season data in local production systems, incredibly important because efficacy varies widely of these products. Practicing good soil health principles only enhances the potential benefits of integrating biologicals into your agricultural system. So any practice that minimizes soil erosion and loss will also impact biological inputs, right? We don't want to be adding in any sort of inputs and having that washed away due to erosion or leaching. Responsible and effective nutrient management, right? So the possibility of these microbes to increase nutrient use efficiency and replace potential fertilizers really hinges on our ability to effectively manage nutrient levels. And then also utilizing these within an integrated pest management system. Okay, and then my final tip is to manage expectations by balancing current practicality with the future's potential. Soil complexity imposes immense scaling challenges, right? We see these relationships between microbes and plants in the greenhouse, and these relationships are critical and so strong, but the scaling challenges related to the complexity of what happens in the field due to microbial establishment, persistence, and competition impose the grand challenge of this. And these dynamics are nearly impossible to measure readily in the field, but we are working on it. Okay, so very briefly, I talked about reading the label, and then reading the label. So the next few slides, I'm going to highlight a handful of labels. These are by no means, this is not an endorsement. These are products that growers in North Carolina have utilized, and just wanted to highlight some things to look for. These were all found online from some Google searches. So this is a dry blended endomycorrhizal seed treatment. We can see we have a guaranteed analysis of 5% calcium, so micronutrient derived from calcium carbonate. We also have some humic acid, which is an amendment, but it is non-food or non-plant food. And then, of course, the complex microbial communities comprised of bacteria and fungi. Here is another biofertilizer. This is a microbial biofertilizer based on a proprietary nitrogen-fixing microbial strain. Right, so this strain is a methylobacterium. This proprietary strain is this particular isolate number, and this is at 10 to the 7th colony forming units per gram. And again, if we look at the application rates, this is a foliar application, and for beans, we want that at 333 grams per acre, applied at the 3-leaf stage. Moving on, some Vault IP Plus. So this is a dual action for disease suppression and also general stimulant. So Vault IP Plus from BASF is a combination of two different components. This first is a mix of 2 strains of bacillus, which are general plant beneficial plant microbes, and then also a bradyrhizobium, which is a classic nodulating nitrogen fixing microbe. And then again, to consider different application rates depending on how many seeds you are treating. So a whirlwind tour of my 10 top tips for using microbial biostimulants. Quick thanks to the North Carolina Soybean Producers Association and their support of my ongoing research. And you can follow some of my results at my my X handle and our extension webpage. Thank you.
Thank you, Mallory. Now we'll hand it off to Laura.
Perfect. Can you guys see my screen okay? I'm assuming that's yes.
Yes.
Okay, awesome. So Mallory did a great job kind of explaining, some of the background of these seed treatments that really feeds nicely into some of the research we've done as part of Science for Success. So this is a trial. We've done 2 years of research across many states. I'm putting my student's name on here, Fabiano Colette. He did a great deal of research and helped me pull together a lot of these slides. So before I talk about the actual research, I want to back up and talk a little bit about Science for Success. So I'm the soybean agronomist at Ohio State University, but I'm part of a network of soybean agronomists called Science for Success. And this initiative is funded by United Soybean Board, but it's to bring soybean agronomists from all over the U.S., land-grant universities, to work together on both collaborative research and extension efforts. And right now there's about 26 soybean agronomists that represent 90% of the US soybean acres. So it's a really nice group of soybean researchers. And so every year we talk about research that we need to conduct to help support our extension programs within our states, but also have a national impact. So a couple years ago, one that really rose to the surface was the use of biostimulant seed treatments. A lot of farmers in our individual states had questions about these products because they were really growing in popularity. So one trial that we just wrapped up was this national screen of biostimulant seed treatments. So you've already seen this figure once, but it is not a surprise that there's just so many products available in this biological landscape. The ones that we focused on for this research were that biostimulant category, which Mallory was also focusing on as well. So we evaluated 9 different biological seed treatments or biostimulant seed treatments over a couple of years. There are some that were extremely commonly seen in products. So Brady rhizobium, Bacillus, Trichoderma, Pseudomonas, Azospirillum. So these ones you'll probably run across a little bit more. There were others that were lesser known, at least lesser known to me. And some of them were quite new, newly released as commercial products. One of them, Methylbacterium hispanicum. There wasn't much published research on it, but I found it was isolated from drinking water in Seville, Spain. And then another one that was isolated from Mexican chili powder. So some ones that you'll run across more, others that maybe you don't see quite as much, but all of these products were commercially available. And so really there's a lot of gaps in the biostimulant seed treatment research. If you look at the published literature in journals, you'll find a lot of studies from Brazil. Brazil does a lot of work in this area. You also see a lot of studies from the Middle East, India, Africa. There are some in the US, but not quite as much compared to other countries. As Mallory mentioned as well, there is, efficacy shown in greenhouse or lab environments, but not as much translated out there into the field. And then also too, do these products work in more stressful environments? So where phosphorus is limiting or there's some type of abiotic stressor. So really we wanted to hone in and try to fill some of these research gaps. And so overall, our objective is just to identify situations where these biostimulant seed treatments could help improve soybean grain yield and then profitability as well. So the first year we did this study was 2022, and you can see the strength of the Science for Success team. We were able to get 17 states involved, and across these 17 states, we had 49 research locations, and we had 6 replications of each treatment. So a very large data set. These are all small plot trials just because of the size. We had to do small plot research trials, but very, very large data set. The 2022 treatment list is here. And so we have these listed by active ingredients, but they were all commercially available. You'll notice some products had one type of microbe, some had mixes of various microbes. Everything was compared to an untreated control, but it was still treated with fungicide and insecticide. So we had fungicide and insecticide on all of our seed, and then we applied those products over top of it according to each company's recommendations. So before we applied these products, we talked to companies and check the labels and make sure they were compatible with the fungicide and insecticide seed treatment. You'll notice, I know I'm from Ohio, but you'll notice we are missing numbers 5 and 6. That's not because I can't count, but there was 2 products we couldn't compare across states in 2022 because they weren't allowed to be used in all the states. And so this goes back to that state level registration. And so there were a handful of products that were not registered in the South, and I think Iowa too had a few that we couldn't use. So there was two products that we had to pull from the larger data set because we couldn't have analysis across all of the states. And this is my students, he pulled from the literature what these different products were marketed for or what their proposed benefits were. So you'll see a lot of things about enhancing early growth, vigor, root mass. Some didn't have a lot of information. Bradyrhizobium, of course, would fix nitrogen from the atmosphere. We had some that were supposed to protect against fungal root diseases, enhance and fixation, improve grain yield, increase crop benefits, which falls into that biostimulant category. The second year of the study was 2023. So we had 21 states. There were some new soybean agronomists that came on board, and they jumped into this trial and conducted research in 2023 with us. And in that year, we had 55 locations across the US. This is our treatment list from 2023. A lot of them are the same for both years, but we had some products that we had to replace from one year to the other year, and that's because there's so much turnover in this market too. So just because we test one thing one year doesn't mean it'll be available the following year. So we had a few new projects that entered into our 2023 study. So just to get an idea of size, when I do in-person extension meetings, I have this big container with beads to kind of give a visual representation of all of the soybean plots we harvested across the US. So in total, with these 2 years, we had 22 states, 104 individual research environments, and in total harvested close to 6,000 individual soybean plots. So if I did research in Ohio over a 2-year study with multiple locations, maybe I'd harvest 240 plots. But with the Science for Success team, we're really able to get a huge amount of data in a very short period of time. So these are the locations we had in 2022 and then again in 2023. Because we know this is such a diverse environment, we worked with a statistician to categorize our different environments based on weather and soil properties. So the different colors and different shapes on this map here are how we clustered our different environments. So Illinois, Ohio, Indiana, Wisconsin, Michigan is this blue area, which is probably most relevant to this webinar, but we did cluster into these environments. So in 2022, there was no statistical difference between our control and the biostimulant seed treatments. And this is by environment. So we have environment 1, 2, 3, and 4. Again, 3 is that blue area, this Midwest region. The black bar is treatment 10. That's our untreated control. And then the different shades of green are our individual biostimulant seed treatments. But across these different environments, we weren't able to see a statistical difference between the biostimulant seed treatments and the non-treated control. 2023 had a similar story. We weren't able to find a statistical difference in soybean yield comparing those individual biostimulant seed treatments and the non-treated control. Again, 3 is that Midwestern region. It's the highest yielding region, which is cool, but no effect of that biostimulant seed treatment. So we worked with a statistician on this because the data set is so incredibly huge. We wanted to make sure we were representing the data well. And so the statistician gave us the best case scenario according to his analysis. And he said the best case scenario would be a 3 bushel per acre yield increase, which is good news. The bad news is it was at about a 25% probability level. So, and that is given the scenario of you can pick the best seed treatment for your operation. So he looked at the best biological or biostimulant seed treatment compared to the non-treated control at each location. And he found a 3 bushel per acre yield increase at 25% of the time. And so when I think about these biological seed treatments, I like to think about the disease triangle as well. I always joke that plant pathologists are contractually obligated to show a disease triangle. So I'm sure you guys have seen this multiple times. But for a disease to occur, you have to have three things in place at the same time. And so you need a susceptible host. So that might be a susceptible soybean plant, a virulent pathogen, so a pathogen that causes a disease, and that conducive environment. So in the case of soybean diseases, usually that means maybe wet conditions or humid conditions. If one of these things doesn't occur, like Ohio this year had a drought, we did not have a conducive environment for disease. We didn't see disease develop. If one of these components is missing, you won't have that disease develop in the plant. So the same can be thought about a symbiotic relationship or a mutual relationship between a soybean plant and a beneficial microbe. You have to have three things in place for that symbiotic relationship to occur. You need to have a soybean plant. You need to have that plant beneficial microbe. And in the case, the ones we were looking at, they needed to be alive. And so they need to be delivered to the seed at a high enough concentration and also be alive. And Mallory gave some great tips on how to keep them alive, which is very important. And then also that conducive environment where they can infect the soybean plant root. And I think the other thing to keep in mind too, there's so many microbes in the soil already. So I'm not a microbiologist, so Mallory could address this more, but there is competition as well with those native species. So 1 teaspoon of soil can have 1 billion individual microscopic cells, which is a lot. And to put it in perspective, in the US, there's about a third of that in terms of the human population. So there's a lot of living activity in the soil already before that microbe is even added. But we do know that a lot of these products can show efficacy in greenhouse situations. So this is from my colleague Manny Singh at Michigan State University. He looked at some of the biostimulant seed treatments we tested in the field, but put them in a greenhouse scenario and withheld water from soybean plants. And the one that was treated looks a lot better, less wilty than the one that was untreated. So again, we can see these show efficacy in a controlled situation, but much more challenging to take that from a controlled situation out into a field scenario. So we didn't see any differences in yield due to the application of those biostimulant seed treatments, but we were able to look at our data since it was so much data and try to figure out, okay, biostimulant seed treatments didn't give us differences in yield. What did? So we worked with the statistician to divide, I guess, our experiments or trials into different clusters based on and divided them into different properties. So what influenced these yield clusters? So I'm just going to walk you through our highest yielding and lowest yielding scenario. So our highest yielding cluster was 77 bushels per acre. So when we look at our large data set, the first thing that divided our clusters was row width. So this arrow here led to higher yields. So in situations where we had row spacing of 15 inches or less, flows down the flow chart this way. Our next way we could divide our data was by seeding rate. And we saw seeding rates that were lower, less than or equal to 150,000 seeds per acre, led to higher yields. And this may not be because of the seeding rate. We do know farmers, if they have a good soil, good field, they'll drop their seeding rate. So this may just be indicative of a nice soil and farmers felt comfortable dropping their seeding rate. And then the final thing that divided our data into this higher yield cluster was year. 2023 was a higher yielding year overall compared to 2023. So our lower yielding cluster is down here. This is 43 bushels per acre. So you can see we had yield variability across the US, which makes sense. So to get to that lower yield cluster, again, our first way our data is divided is by row width. So we flow this way now. And these were row widths that were greater than 15 inches. The next way our data is divided is by cluster. So we had those 4 different environmental clusters. Basically, if you're not prime in the Midwest, you had a lower yields, which I think also makes sense. And then the final way we could divide our data was by previous crop. We saw previous crops of cotton and wheat lower yielding than other previous crops. So we can even though there was no statistical difference with these biostimulant seed treatments, we were able to see how other practices at these research locations may influence soybean yield in general. So with that, I just want to thank, the Science for Success team for working together to pull off this study. As far as I know, it's probably the biggest biostimulant seed treatment study available right now. And also United Soybean Board and USDA co-funded this project. So we got funding from both to do this research. So I appreciate your time, and I think we can open up for questions if there are any.
Thank you so much. I know that Mallory, thank you so much for answering some of those questions already. And I think we have one for you, Laura. For the small plot trials, do you have any information on the fertilization?
So we have that information. So we had, over 100 different environments. So each state does things a little bit different. We do have baseline soil data for all of the fields. So we had each collaborator pull a soil sample. So we have all the baseline fertility of phosphorus, potassium, CEC. And then the fertilizer practices, we just ask people to do whatever is locally appropriate and needed in their field situation.
Okay, thank you. We had a lot of great questions come in. So thank you so much before the webinar. So I'm going to start going through some of those. And I can give a perspective from Illinois if that helps. But overall, someone asked, how common is just seed treatment in general among large-scale grain growers or across, I would say across the, farmers across the U.S.? Do you guys want to go ahead and comment for your state?
In terms of just seed treatment is probably nearly everyone in terms of fungicide, insecticide, and that's a fairly standard. The addition of those biostimulants, I would say there's probably a fairly good portion of farmers using it, especially Bradyrhizobium japonicum, which is kind of the standard biostimulant. I don't know if I want to pull a number out. I've talked to farmers and they say that they do it if it's easy. If it's something that's already applied to their seed, or the person they're purchasing the seed from has it on, they can do it easily, they will do it. But if it's something that takes time out of their day or their operation in the spring, then they may not do it. So it's variable.
Mallory, do you want to comment, or you don't have to?
Yeah, no, similar. I think the standard fungicide, herbicide, insecticide treatments are pretty, right, like most, almost all. And then seed treatments vary. I know there's a lot of products that are in furrow treatment, but that requires specific equipment. But I think if growers have that equipment, they tend to gravitate towards those applications as well.
So Illinois, I would say, is one of the top states to have seed treatment in general on soybeans. And again, a lot of them do the fungicide, insecticide package. And there's a lot of seed companies that offer a general, just that's what you get whenever you order the seed if it's upstream treated. But we have a very high amount of downstream treated seed, meaning for example, the dealer treats it on farm. And so then the farmer is able to maybe pick the package that they would like or components that they would like on the seed. So if we have any other questions on that, we can elaborate more. We have several questions. I don't even know where to go. Okay. Is there any way to determine how long these substances or organisms survive in the field, or if they even survive being applied? Who wants to take that one?
Mallory is a microbiologist, so I will
Yeah.
let that.
So and this follows up after there was maybe a similar question that I answered in the chat. Technically, yes. Feasibly, absolutely not. The just massive cell complexity of soil and all of the microbes living in there make it nearly impossible to track establishment. I mean, there's a few studies, a few products that have published some work demonstrating that the product is present and colonizing plant tissues in the greenhouse and potentially in the field. But just for logistical, methodological reasons, it's not possible.
And I had another question, Mallory. You had answered one question that kind of spurred another question in my mind. So if you have multiple organisms or microbes in a jug, for example, and it's at the warehouse, and by the time it gets to the farm, is it possible that different one microbe might take over? Like it could be a different or populations within the jug, or is that not possible?
Yeah, absolutely. It's really challenging to grow mixed communities of microbes in liquid. Um, so technically difficult. Many of these products, though, are taking advantage of microbial dormancy, right, or sort of low state of metabolic activity. So that's why, we often want, if you have liquid formulation, sometimes the recommendation is to refrigerate at 4 degrees, so microbes aren't really active. So it's less likely that they're growing and dividing in the jug, and we want that to happen once they're applied in the field. But no, those dynamics are always playing out. There usually will be a dominant winner if a mixed community of microbes is actively growing.
Interesting. So Lewis, actually, these are two questions I think that were submitted ahead of time. So he's wanting to know if you are familiar with this particular microbe or yes, or product there. Are either of you familiar with it? Or can you see it in the question and answer box? Sorry.
Yeah, no, I'm not familiar with this specific product, although Bacillus again is another microbe well known for plant beneficial relationships. When I was just talking about dormancy, so Bacillus are also have these strategies where they can go dormant, therefore sort of withstand being dried down into a powder. I'm not sure what this product is, but certainly a strong contender for what a lot of microbial inoculants are these days.
And then I think they're using it to reduce fusarium species on small grain crops. Any comments on fusarium? And I could make some comments too, Laura or Mallory, on fusarium affecting soybean crops. And is it a major economic threat?
Yeah, I'm glad I got the question ahead of time because I was able to do some poking. So, there's a lot in that fusarium category, I guess. But the one that I saw from the Crop Protection Network, I was looking at their most popular diseases or commonly seen diseases in certain years, and soybean sun death syndrome was at the top of that list, which is caused by Fusarium vigilifolium. So yes, the crop or the plant pathologists have identified that as a top problem and yield reducer in the disease arena for soybeans. And if you have, yeah, I'd like to hear your comments too, Stephanie.
Yeah, Mallory, do you have anything to add or?
Negative. I'm not a plant pathologist, and I would reach out to my fantastic NC State pathologist colleagues.
I know, I totally agree. Sun death syndrome is a major issue here in Illinois as well, and especially on our own farm. That was one of the first diseases that I learned about because it was not good on our farm. But there's, I don't know, there's thousands of different fusarium species out there. And from my experience, in the last year or so, it seems like we try to send off some samples if there's an issue out in the field. And a lot of times you get several different pathogens. And so I've gotten questions on it. And oftentimes it'll say fusarium species, or, depending on who, if they break it down to exact species, I don't know if they do or not, depending on the plant clinic or whoever they're sending it to. But in my experience, so thus far, and I'm not saying it can't change, is there's usually some other kind of stress. And I can give out some examples, and it's sometimes we need to do a little bit more homework. I usually go ahead and pull a soil sample. I pull a soybean cyst nematode sample. There oftentimes is something else going on. For example, water, sitting in water for a while. We've had that issue, and it's compacted and it sits in water. I've sent these samples off, and you get a fusarium. So I would say in Illinois, from my experience, it's often a secondary or like a red flag pathogen that something else is going on. But I'm not saying there are in other areas that there's fusarium that could be a major pathogen on a plant or soybean. So next up, let's see. Do you think we will ever see some substantial yield gain from any of these products, or a new, maybe like a upcoming product? And are they worth the cost? Does anybody want to take that? And you do not have to take that question if you do not want to.
Yeah, I mean, in our Science for Success study, we didn't see any significant difference between that untreated control and the biostimulant seed treatments. But there are, Brady rhizobium does work. If you have a field that has not ever seen soybean or hasn't seen soybean in a really long time, you do need to inoculate, and you will see a benefit from having that Brady rhizobium japonicum inoculation on your seed if it's a new soybean field. So, that's, I don't want to say the standard, that's the most commonly used, and it does work. If you don't have a history of soybean, that product does work. And I know there was questions about reducing your fertilizer, and the answer is yes. If you have Brady rhizobium that's successfully nodulating your soybeans, that means you don't have to apply nitrogen. And so I think maybe that gets forgotten too. There is a product that does work, and it does allow you as a farmer to save costs by not applying any nitrogen to your soybean.
Yeah, I'll just
go
add,
ahead. Sorry, go ahead.
So for many generations, we've been breeding crops for yield enhancement. So the biggest way to get better yield is through plant genetics, right? So like variety selection, and certainly like fertilizer and your production system. With the inclusion of biostimulants, I think one area that is incredibly promising is under high stressed conditions, right? So we do have some compelling evidence of field trials where biostimulants are alleviating salt or drought stress, a significant yield increase. However, again, the caveat though is if that yield increase is economically viable or relevant, right? So if we have whatever, a 12% yield increase on, a tiny little salt sick plant, it's not still super economically viable, but I hope that is the direction that we're going with innovating these products of abiotic stress tolerance and also reducing fertilizer inputs.
Thank you. Thank you so much. So I don't think I have any other questions. I hope I hit most of them that were sitting ahead of time. But if there's no more questions, then we're going to go ahead and wrap up. And I just want to thank you both so much for taking time out of both of your busy schedules to be with us. It sounds like you're sheltered down from the weather, even in Ohio and North Carolina. So thank you so much, and thank you to the attendees. We really appreciate you coming here. If, as always, the recording is available for this webinar, and you can always subscribe to Field Advisor, and that's where these webinars are actually held. If you go to the webinar section on Field Advisor, you can subscribe also to the e-newsletter, YouTube, podcast. We're also on Facebook as well as X and Instagram. And so lastly, the next Field Advisor webinar will be on red crown rot, and it's going to be an update. And this is going to be held on February 20th at 10 a.m. And so stay tuned, more details to come. So if you want to get more details, always subscribe to Field Advisor to get notified of the dates of all our webinars and topics and times. And again, we'll have a live registration for those webinars, but they'll also be, again, recorded so you can have the slides later on if you would like. Okay, thank you so much.